Hello everyone and welcome to my channel Easy Life Science. Here I am talking about bacterial conjugation. So please subscribe my channel Easy Life Science, like my video and share it to your friends. Don't forget to press the bell icon so that you will get the notification of my newly uploaded video. So let's start. The bacterial conjugation is a process of a gene transfer. It is also called as a horizontal recombination. It is a process of a genetic transfer from a donor cell to a recipient cell when they are physical contact with one another. Bacterial conjugation is also known as a type 4th secretion system. The conjugation occurs in between a number of bacteria including gram-positive bacteria and gram-negative bacteria, and even occurs between bacteria and plants. The process of conjugation, discovered by the two scientists Joshua Lederberg and Edward Tatum in 1946. They experimented with two exotropic strains of E. coli K12. The exotropic strains are mutated for one or more genes, so that the gene products are does not formed by these organisms. For example, if the methionine gene is lost, then the organism cannot form a methionine or cannot synthesize a methionine. So on a minimal media in which there is a absence of a methionine, such cells cannot grow. It means that the exotropic strains are mutated for genes which are responsible for a production of a particular amino acid or vitamins, and these exotropic strains does not grow on a minimal media, whereas the prototropic strains are with intact genotype and they grows on a minimal media. For the experiment, the scientists Joshua Lederberg and Edward Tatum made a use of E. coli K12 strains. Two exotropic strains of E. coli K12 were used, strain A or strain B, or strain first or strain second. The strain A and strain B were separately plated on a separate minimal media and incubated at overnight, and no growth were observed. When strain A and strain B were mixed together and plated on a minimal media, resulted in a prototropes and colony were observed. It showed that somewhat there, due to which the strain A and strain B grown on a media. The experiment carried out by the Lederberg and Tatum. The strain A, the genotype of strain A are methionine negative, biotin negative, threonine positive, leucine positive and thiamine positive. The methionine negative and biotin negative, it means the genes for synthesis of methionine and biotin are lost by a mutation. So strain A does not grow on a minimal media which lacks a methionine and a biotin, whereas a strain B, genotype of it methionine positive, biotin positive, threonine, leucine and thiamine negative. It means that the genes for synthesis of amino acid like threonine, leucine and thiamine are lost in strain B. So this strain also does not grow on a minimal media. But when the scientists mixed these two strains in one tube, after mixing, the cells were plated on a minimal media and growth were observed. The scientists Joshua Lederberg and Tatum does not give a evidence for a cell-to-cell physical contact required for a conjugation. For this, the scientist Bernard Davis in 1950 carried out one experiment and showed that there is a need of a physical contact to a conjugation, the Davis placed each strain in one of the two arms of a U tube or a Davis tube, separated by one filter or one fritted glass filter. Prototropes was not observed in either of the two arms of the tube, and later he given that for a conjugation there should be a physical contact. The mechanism of F plus and F minus conjugation. In a conjugation, one cell acts as a donor or a male, while another as a recipient or a female. Maleness in a bacteria is due to presence of a transmissible extrachromosomal genetic element termed as a plasmid, and this plasmid is called as a sex factor or a F factor or a fertility factor. The cells possessing free F factor are called as a F plus cell. The cells lacking F factor are called as a F minus cells. F factor may remain free as a extrachromosomal, or it may get integrated into a chromosome. The cell in which F factor integrates into the bacterial chromosome are called as a HFR cell, high frequency recombinant cell. Some property of F factor or a fertility factor are mentioned here. The F factor is a extrachromosomal transmissible circular DNA molecule. It is autonomously replicating DNA molecule. It does not depend upon the chromosome of a host. It remains free in the cytoplasm or occasionally get integrated into a host cell chromosome. Each cell usually contains one F factor. It governs its own transfer to another cell. It also mediates transfer of host chromosome and other plasmids. It gives a surface property like formation of 6 pili. The size of F factor is 40 times less than that of the size of host cell chromosome. It is double-stranded DNA of about 30 mm length, having 1 lakh nucleotide base pairs. It consists of TRA genes. The TRA genes are involved in a transfer of F factor into a female cell. 19 transfer cells are there, or 19 TRA genes are there in a F factor. The F plus and F minus conjugation or a cross between F plus and F minus cell. The F plus cell contains a F factor carrying the genes for a sex plus formation and a plasmid transfer. One or more F pili are produced by each F plus cell. The sex plus specifically binds to a protein, that is a outer membrane protein A, OMPA protein of F minus cells, thereby initiating a transfer replication and the process of gene transfer by a conjugation. Donor cell produces a pili. The pili attaches to a recipient cell, and one conjugation bridge is formed between two cells. Only a single strand of a plasmid is transferred to a recipient cell, and complementary strand is formed on that single strand. In this image, there is a cross in between F plus and F minus cell. This is a genome of a donor cell or a F plus cell. This is a genome or a chromosome of F negative cell or a recipient cell. This is a plasmid or a F factor. The one strand of a plasmid DNA is broken at a site ORT, that is the origin of transfer, and it replicates by a rolling circular mechanism. The 5' end of the broken strand is displaced and transferred to a F- cell. The channel for DNA transfer could be either a partial conjugation bridge provided by a refraction of a F plus or a hollow F plus. The cross between two F strains does not produce a recombinant. In a F plus and F minus cross, one copy of F factor received by a F minus cell, while another copy remains in a F plus cell. Thus, the F minus cell converted to a F plus cell. That means a donor cell remains as a donor, but the recipient cell gets converted as a donor. F minus cell becomes a F plus because of this cross. Then the HFR conjugation, or a high frequency recombination conjugation. In 1950, the scientist Luca discovered a strain of E. coli that was very efficient in transferring a chromosomal DNA, and he designed this strain as a HFR. The HFR strain is derived from F plus strains. In F plus cells, the F factor lies in a cytoplasm, and only rarely it combines with a bacterial chromosome. The cells in which the F factor integrates into a bacterial chromosome are called as a HFR. The cells are called HFR because they show a very high efficiency of chromosomal gene transfer comparison with F plus cells. In a conjugation between HFR cell and F minus cell, HFR is very high, but the frequency of transfer of a whole F factor is very low. The integration between the F factor and a chromosome occurs at a site where a specific sequence are present. There are 4 such sequences on a F factor and more than 20 on a E. coli chromosome. Integration between F factor and a chromosome occurs by a pairing between a homologous insertion sequence. This is a formation of HFR cell. The F plus cell, it is with a F factor or a plasmid. The plasmid gets integrated into a host cell chromosome and forms a HFR cell, and this is a HFR cell, high frequency of recombination. The cross between HFR cell and F negative cell. In this cross, the F factor directs the synthesis of a F villi, carry out a rolling circle mechanism, and transfers the chromosomal genes to a F negative cell or a recipient cell, that is in this experiment to the strain B. Actually, the experiment carried out by the scientists Liederberg and Joshua. Scientists Liederberg and Tatum. The donor cell was a HFR and a recipient cell was a F minus cell. HFR cell, high frequency of recombination. In this cross, the HFR cells shows a presence of a F factor. It directs the synthesis of a F villi, carry out a rolling circle replication, and transfers the chromosomal genes to a F negative or a recipient cell or to the strain B. Gene, for example, threonine, leucine, and thiamine. In this cross, here is a transfer of a chromosome genes from a donor cell, that means from a strain A cells to the F minus recipient cell or a strain B. Chromosomal genes that are transformed from strain A to strain B are genes for threonine, leucine, and a thiamine, which are absent in a or which are deleted in a recipient B strain. Entering in a recipient cell, the strain B, the HFR then integrate into a recipient cell chromosome and leaves a gene of a donor cell, that means genes from a strain A like threonine, leucine, and thiamine in a chromosome of a strain B. Thus, when such recombinant strain B divides, shows a prototypes like methionine positive, biotin positive, threonine positive, leucine positive, and thiamine positive, and grows on a minimal median. This recombinant cell or a B cell further becomes as a donor cell and also donates its HFR to a other exotropic cell. So many of a cell in a mixture becomes a prototropic and grows on a minimal median. In this way, the HFR and F minus cross occurs. Then F prime, the F prime or a sexduction or a F duction, integration of a F factor in a donor bacterial chromosome is reversible. It may detach from a chromosome. Detachment of a factor from HFR chromosome at a site away from a site of integration leads to formation of a F factor containing a segment of bacterial chromosome. Such F factor is called as a substituted F factor or a F prime. The F prime can F duces or a sex duces to a F minus cell for specific genes that are carried by. For example, F prime lac positive donor cell transfers a lac positive gene to a lac negative recipient cell and convert it into a lac positive. This process is called as a sexduction or a F duction. In general, a gram negative bacterium carrying a conjugation plasmid can mate with a wide variety of other gram negative bacteria and transfer plasmid as well as chromosomal DNA. This mating may be interspecific in between E. coli and E. coli, or intergeneric in between E. coli to Proteus mirabilis, E. coli to Salmonella typhosa, or in between Salmonella typhosa to Proteus mirabilis. This is a conjugation in between F prime and F minus cell. Here in this image, three crosses are there: a cross between F plus and F minus cell, cross between HFR and F minus cell, a cross in between F prime and F minus cell. Here is one summarization: a cross in between F plus and F minus cell, a cross in between HFR and F minus cell, and a cross in between F prime and F minus cell. So this is about the conjugation or a bacterial conjugation. Thank you for watching my video. If you like my video, subscribe my channel Easy Life Science, share the video to your friends, and don't forget to press the bell icon so you will get a notification of my newly uploaded video.